It is my pleasure to introduce Marcin from Takeda. He's the Director of Data Integration and Analytics. He has over 20 years of experience executing multidisciplinary translational projects, pioneering research in the analysis of clinical, molecular, and genomic data for drug discovery in rare and also common diseases, and precision medicine. So without further ado, I'd like to introduce Marcin, and he'll be speaking to us today about target identification and validation with FAIR Genomics data lakes. So thank you, Marcin.
Thank you so much. Hello, everyone. First of all, I'm just a messenger. A huge number of people is behind this project. And here's the authors only from Takeda that are currently working on this one. Additional people who work on this project is Chandra Salma and his team from San Diego. They were instrumental. Chandra Salma was a visionary for this project, and he basically made the landscape, how this should be executed. Additional thank you to Zifo's team, to Ashfarya, Tran, and Raghavi, and obviously the developers team in India that without coders, at the end, this wouldn't be existing at all. So thank you so much for everyone who contributed to this amazing project. That's the disclosures about the authors. And the target identification and validation is kind of crucial step, as we all know, in drug development cycle. If you know the right target, it usually helps you to understand the disease, which kind of sounds weird because you thought that first you understand the disease and then you identify the targets. But very often, pharmaceutical companies try to guess the target. And then when you luckily guess the right target, you can better understand the disease. And when you have this target identified correctly, it helps you to identify how you can do a druggable of this target, how to do a rational drug design, and then it can speed up the entire process of drug development because you don't need to repeat the cycles if you miss the target at the beginning. And it can also lead to the personal medicine solutions. And probably most of you know this extremely very well, so apologies that I'm basically repeating the knowledge that is widely known. But many drugs in the past and still right now are failing not because the drug was not supposed to do what it's supposed to do. It was exactly doing how it was designed, but the target was incorrectly identified. Very often because of the not understanding the biology, which is complex, obviously, or because of the off-target effects and being more precise about the therapeutic window. So even if your target is associated with the disease, but maybe the impact of inhibiting, for example, this target might not be so big on the disease. And if it has no big enough for disease, then you're trying to boost the concentration of your drug to inhibit the target even stronger, and then your therapeutic window becomes shorter because you always have some side effects from any medicine, or even the stuff like water. If you put too much, you will have side effects. And very often, if you have even the good drug target, but there is no biomarkers or preclinical models, then it's very difficult to work on developing the drug because at least there's always these triangles or maybe even the square drug target indication, biomarkers, and good to have the good preclinical models for the development. And in the numbers, it looks basically like that. You can see on this lower panel that the probability of hitting the right target is still on the level of kind of few percent. And then on each other steps, the percentage of the successful rate is significantly higher. But this means that you have a high chance to make an error in early stage, and this error is basically being propagated to the end of the cycle development of drugs. So that's why it's so important to hit the right target at the beginning. And that was the whole vision behind, we call this one preclinical and translational science PTS digital efforts that has been recently promoted to the one research digital efforts. And the whole vision is to have those FAIR data lakes with harmonized metadata and data governance that could help us to apply the AI ML on top of this one. But the biggest underlines goes for the data verification, we are always saying that we need to eat our vegetables first. So first we need to verify the data and harmonize this correctly. And then we have a tasty cake after the dinner, which is going to be applying AI ML. So the data verification first, and then let's enjoy and play with the AI ML on top of this. Each of the data lake is kind of presented independently, because this is what we are doing. We are picking up different technical solutions for each different data type. So there's no kind of concept of one data lake. It's a concept of one data mesh. But each of the data lake has its own technical solutions for different data types. For genomics data, we're currently using TileDB as an example. For the clinical data, Zifo built for us a clinical data lake, which is non-GXP clinical data lake. It was kind of very, very important for us to find the balance. Like, we didn't work to operate with the clinical GXP environment when you are not allowed to do many, many things. We really wanted to grab the data outside of this GXP environment and put to the non-GXP environment. This is a kind of perfect balance, because the clinical team always have an allergic reaction about whenever you wanted to touch clinical data. And we understand why, because if you find something, it might affect the entire process. You need to report it, and then 2 years later, you can prove it that it was just data artifacts. There was nothing worried to be about the data. So that's why it's important to have those two independent clinical data environments, GXP and non-GXP. And this is what we manage here. But coming back to this vision of this one data mesh, so despite the fact there are multiple data lakes independently solving purpose of different tasks, but everything is connected together as one data model. So different data lakes, but one data model, one data mesh, and one data governance. This one data governance is also very, very crucial to fulfill that the clinical team will not have allergic reaction, because the right people have access to the right time to the right data. And it's not about only whether you are qualified or not qualified to understand the data and to make a decision, but in the case of the clinical data and the genomics data that, for example, coming from the different biobanks, like UK Biobank, it's also about what kind of questions you wanted to ask. We have to have in our head in mind that this is like a real people's data, mine, yours, and we are agreeing to share this data under the certain rule what kind of question will be explored on top of our data. So this has to be also be incorporated in this entire process. And as you can see from this picture, this is very complementary what Mike was talking about the CMC, how important it is to make those connections together to then start applying those machine learning and AI on top of this one later on. And because this is extremely Herculean task to do, this is what Shandor envisioned. He envisioned that we will basically split this to the DPs, to the demonstration project. And each of the demonstration project will be focused on different aspects of the problem. So we are not only building a plane, but right away you wanted to fly with this plane by focusing on particular aspects. Maybe not all of the possible routes are right away available, but at least some of them. And one of those demonstration projects is demonstration project number 1, that is basically focused how we can take the patient's clinical data and the patient genetics data, and how we can grab the variants from the genomics data lake, extract those variants and see which of them affects the protein structure, and then color code the protein structure with the patient's phenotype. So this is extremely cool stuff, because we simultaneously joining together patient genomics data, clinical data, and external public domain data sets in one application. And we know this already from the cancer world, that the variants affecting the amino acids change in cancer, they are collocated together in the three-dimensional space. It was already nicely shown almost a decade ago, but right now it's amazing to have this working tool right away when you can search through all of the proteins. Because this is what is done right now. We have the predicted protein structures for most of the human proteins. These models might not be 100% accurate in entire space, but very multiple domains of the human proteins are are predicted with the huge accuracy. And I wouldn't give this talk today without this one. It's a perfect timing. Like a week ago, there was a Nobel Prize for David Baker's and for the people from the DeepMind in Google's for how we can predict the protein structure. And David Baker was a pioneer in predicting protein structures and also designing artificial proteins. And I have a privilege to work in 2002 in David Baker's lab when he was working on this top 7. That was the name of the first artificial proteins of over 90 amino acid long that was designed. And then my colleagues and I, when David published his paper in Science, we wrote a letter to the Science editor, highlighting how challenging it was, because that time was extremely difficult to predict the protein structure de novo if you don't have a close homologs, and you don't have it when you're starting to predict and design artificial proteins. So knowing that now we have all of those proteins, human proteins, with basically predicted protein structure, and for many of those we have already crystallographic structures, we can map the patient's variants on the 3D structure of the proteins and ask the questions how they are colocalized three-dimensional space, and maybe this could basically not only identify future targets, but additionally suggest the mechanism of action which speed up the entire process of understanding the biology and the rational drug design process. And this is what Zifo helps us to basically build. We build this clinical and genomics data lake with the graphic user interface. So we are uploading a clinical data, you are uploading a genomics data, then you make the integration of the clinical and genomics data. You take the protein structure from the PDB or from the UniProt, in the case of the AlphaFold 2 predicted models, and then you have a visualization. How this looks like under hood. So going from the left to right, you take the VCF files, you take the clinical data in ESDTM format, you harmonize this, you're taking the protein data bank crystal structures, or you're taking AlphaFold 2 predicted models, you're loading this on the fly, and then from the user perspective, you basically ask the questions, okay, this is my study, this is my gene of interest, do a visualization. And what was previous speaker was mentioned, the verification is extremely important, okay? Why we don't have still a quantitative measurements, how to say how fair we are doing, but if we have kind of qualitative quantifying way that we basically asking Zifo's team members and our team members to do this manual evaluation, and then we are basically comparing score between different people. How fair is our data in the genomics and in clinical data, in the clinical data lake? And so we have this long list of different questions. Is this one accomplished, and how it is accomplished? And each of the person gives its own score. So when it's still not automatic way, but despite the fact it's manual, it gives you the overall picture how good we are doing. And I wanted to mention that this is how interoperability we achieve relatively high scores, and that was both the Zifo's team and our team recognized this one, was thanks to side-by tools, the Sentry, the Termite, and the Workbench. We basically use those tools on top of our SDTM ETL engine, which basically speed up the entire process of making sure that we are using a standardized vocabulary, and this is nicely interoperable between different data sets. This is what is in the progress, is to make sure that our findability and being more precise global unique identifiers and being even more precise global unique persistent and resolvable unifiers are correctly implemented. We are starting using Accurates to help us in this process. And this vision comes from the AstraZeneca to start using your global unique identifiers as URLs. And the vision of using the URLs, it's so beautiful because if you have any paper or any small molecules or any anything else, and you are wondering, is this identifiers been changed or not changed, then you can go to any browser, you can just type it, and then if it was changed, you're going to be basically redirected. And you see the whole history, how this, for example, a small molecules its global unique identifiers was changed during the time. It started from this one because it was not considered as a therapeutics, and then it was promoted to the tag XXX. And with this vision that everyone from its browser can see how is the life of these identifiers and where the current version of the identifier sits on is a perfect solutions for us. Now how this looks like from the user perspective. From the user perspective, we have a nice graphic user interface. All of this, what you will be seeing here is or the dummy data or a public domain data, obviously, but just to, if you see some weird numbers, I'm just explaining why they are so weird. So you go to this beautiful graphic user interface, you select your study, and then you automatically are being connected to the clinical data lake, and then you can extract the patient's data, and you can start filtering the patients by different properties to narrowing down which list of the sub-cohorts you are interested. And when you selected your patient, then you go into the next one step. And the next one step that you don't see here, unfortunately, but in the next one step, simultaneously, you see all of the patients' genomics variants, plus the public available variants for selected genes. So you have selected the genes, you have a selected patients. Automatically, from genomics data lake, you're extracting the patient's variant, and you would see them on the top. And then on the bottom, you would see all of the public domain variants. And then again, you can start applying filtering that you are only interested in the variants that has a SIFT score with a certain range. Or you can select manually some of the variants. And then you can take this data to the viewer. The public domain variants are shown in gray. And in color, you would see the patient variants, and the beauty is that we are coloring the patient variants, how they are phased or not phased on different strain of the chromosome. This is a genomics view from the perspective of the proteins, how the particular position is changed to which of the amino acids, but you can also switch the view on the patient level data, then each of the row would be a patient level data. And then in the next one step, the last one, you could take this to the protein structure. Again, in the colors, you would see the patient's data that you can color by the phenotype. So it's like, okay, give me all of the patients that has severe type of the disease versus the patient that have a less severe type of the disease. And then the vision is that they would be colocalized those amino acids that corresponds to the variants from those severe patients would be colocalized in the three-dimensional space of the proteins. And you can simultaneously ask about the common variants. Are the variants that change the amino acids that are extremely rare in a nomad, are they also colocalized in this area, which additionally may suggest that indeed those variants and that corresponds to the amino acids might explain the disease and the mechanisms of actions. And are the common variants that we can see everywhere, maybe they are in the part of the domain. Thank you so much. I'm happy to answer any question.
Questions? Everybody wants coffee, I know.
Yeah, I know. This happens when you're at the last one before the break.
Hey.
I'll give you a quick one. So you mentioned that you have one mesh and multiple data lakes. Can you tell me about your data lakes? Are these external data lakes from a Snowflake or Databricks, or are these things that you built on your own purpose focus?
Perfect questions. As I mentioned, we are basically cherry-picking different technical solutions. So yes, we're doing a Databricks if something is not solved already. But we always wanted to take something off the shelf. Is it already available? So for the genomics data, we're using, as example, TileDB because it was already available. So technically speaking, we could develop our own stuff within the Databricks. But because luckily for us, it was already available as a TileDB, then we are using TileDB. We have already the entire user interface and APIs to upload the data and so on. And the second example that is not part of this today's talk is Graph Database. We are using OntoForce Discover platform for Graph Database. Again, we could do this by ourselves using an Neo4j, but because it was already available, and not only the infrastructure with the graphic user interface arrives to this data mesh, but it also comes with the public available data being already harmonized, an ETL engine that allows you to upload your own and the licensed data for automatic data integration. So those data lakes concepts are different technical solutions for different data types. We always try to take off the shelves already available stuff. We are not Broad Institute. We are a pharmaceutical company. We don't want to design something that others can do better and faster than we are. And yeah. Thank you so much.